Welcome to Space Biology today, Series 26, Growing Plants in Space. I'm super excited to talk to you about having real food in space. So in this episode, part 1, we're going to learn how to grow plants in space with Dr. Natasha Sng. She's a NASA space biologist. And in the second part of this show, we will learn from Prof.Dr. John Kiss. He's a space biologist and a dean of the College of Arts and Science at the University of North Carolina-Greensboro. A presentation about plants that we need to go to Mars. In the second part of this show, we will have a debate on how spaceflight environment is affecting plants, how to grow and thrive, which new systems should be optimized for performing plant life and space farming, which are the effects of chronic exposure to cosmic radiation on plants. So welcome to our distinguished guests today. Dr. Natasha Sng is a NASA space biology postdoctoral fellow at the University of Florida, where she works on investigating the plant microbiome for spaceflight application. She did her doctoral studies at the University of Florida Space Plants Laboratory, where she worked on projects that studied the effects on spaceflight on plant biology. Welcome, Natasha.
Hi.
So let's share with us how we grow plants in space.
All right. So since the Space for Women show is a program to kind of facilitate access to space biology for women and girls worldwide, I thought I might share my story with you today on my journey, basically. And I hope that maybe my journey might inspire, someone in a similar situation to essentially just chase their passion and pursue a career in science and technology within the space arena. All right, so I grew up in Singapore. And growing up, I always wanted to be an astronaut. I remember writing my first childhood ambition essay at the age of 9 years old. And I remember writing it about how I really wanted to be an astronaut. And I think if I were to be honest, I kept that ambition all the way till I was 16. In Singapore, when you turn 16, you finish your secondary school education. And then you kind of have to make a hard decision on what kind of career you want to pursue. And so I remember talking to my family and my friends, and they were all really supportive and very encouraging, but they were also realists. So Singapore doesn't have a space program at that time and any kind of space-related research. So I had to decide something else that I was interested in. And in school, I really liked science a lot and I did pretty good. So I decided that, you know what, I'm going to pursue a career in science. And after my secondary school education, I went to the Polytechnic and did my diploma in biotechnology. And then I think that's where I knew I was on the right track. I found my calling. I really enjoyed everything I learned and I was really curious, enthusiastic about the subject. So yeah, so then I pursued all the path towards becoming a scientist. And so I'm gonna share with you some of the pivotal moments in my academic career and especially highlight the fantastic mentors that have helped me get where I am today. All right. So it all started when I met Dan. I was working at the Cancer Science Institute of Singapore, and Dan was the director over there. And one summer, his daughter was doing an internship in the lab, and I was in, and so he took us all out for lunch. And Dan is the kind of guy that knows how to motivate and inspire students. You know, if you're curious about something, he would kind of urge you to go find out more and help you get to where you need to be as a student. You know, and so he sat me down one day during lunch, and we were talking, and he was like, "So, what drives you? What are you passionate about?" So I told him my childhood ambition, and he was super supportive. He was like, "You should definitely pursue it. You need to go to the US." And I was like, I was maybe 19, 20, and I was, but eventually he helped me get an internship at Harvard in the US, and it's a 3-month internship, and that's where I met Susumu. So Susumu was a really fantastic mentor. He invested a lot of his time and effort and really bolstered my confidence in research. So then my 3-month internship became a 2-year master's thesis. And to be honest, I don't think my parents were really happy because it was supposed to be a short-term thing, and then it led up to like a lifelong change kind of thing. But I think now that they have their 2 grandkids, they're pretty happy about it. But anyway, so again, after my thesis, he sat me down and he asked me what I was passionate about. I told him I really love the science that I'm doing, but it wasn't like fulfilling um, everything I wanted. So there was something kind of missing. So I told him my interest. And again, he was really supportive. He told me that, life is too short not to pursue your passion. So he kind of helped drive that desire in me again to like, okay, I'm going to do something with space biology. So then that's where I begin the next chapter of my education. I met Rob and Annalisa at the University of Florida. Again, they were both wonderful, brilliant mentors that provided me with so many opportunities and great experiences. One of these great experience was to participate in a parabolic flight campaign. So in that campaign, I actually got to experience weightlessness, which was incredible. So I'm really thankful for that opportunity, and I don't know if they know how much that meant to me, from wanting to be an astronaut to being able to experience weightlessness is just incredible. So in their lab, and that's where I basically begin my journey in space biology. What they do in their lab is they focus a lot on trying to understand the basic molecular response of plants in the spaceflight environment. Kind of like what some of the gravity mediated responses are, and how do plants basically cope in an environment that is outside their evolutionary history. What genes are turned up and what genes are turned down in order to help the plants adapt to the spaceflight environment. Very, very molecular based. Okay, so here's an example of how a spaceflight experiment is conducted in their lab. So what we do is we have petri dishes like this square box over here, and we plant seeds, like these little seeds over here, onto these fighter gel plates. We call these fighter gel plates because it has all the necessary nutrients and water for plants to thrive. The plates and the seeds are then wrapped up in a cloth. This is to prevent the seeds from being exposed to light. In this instance, light is the cue for the seeds to start germinating. and we don't want plants to germinate before they get onto station. So the wrapped seeds, the wrapped plates are then transported to the ISS. In this particular experiment, the seeds were transported on the shuttle STS-129. I believe it's back in 2009, 2010. And once on board the ISS, the plants are unwrapped by the astronauts and they are put into a growth hardware so that images can be taken and light is being exposed to them. So the seeds germinate and the plants grow. And once they've grown for a desired amount of time, then the astronauts will harvest them. In this picture, you see astronaut Jeff Williams harvesting our plants in a device called the KFT. You see the KFT over here, and that's the Kennedy fixation tube. It fixes plants in orbit so that we can study the genes that are expressed in that environment. The samples are then brought back to Earth and then sent back to the lab, and all us happy scientists in the lab start extracting RNA and try to understand the gene expression patterns of the plants grown in space. All right, so then the main question becomes like, how do plants behave in this unique environment? One of the interesting things that Rob and Annalise's group have found was that every organ in the plant, so the leaves, the hypocotyls, and the roots, they all respond very differently. Here in red, these bars over here represent, each single bar represents a single gene. And the red color basically tells you that the genes are turned up. So we call this upregulated in space. And those that are green basically are turned down. So they're downregulated in space. And this is basically a space experiment compared to plants grown on the ground. So like we have ground controls and stuff. So we know exactly what's going on in the space flight environment. Yeah, so it's very interesting. You can see over here, there is a bunch of genes that are highly downregulated in the hypocotyls, but in certain scenarios are upregulated in the leaves and the roots. So the other interesting thing that was found within the scientific community was the genes that changed in response to the spaceflight environment. They can be grouped into the following categories: cell wall remodeling, reactive oxygen species signaling, redox signaling. These are some of the categories that we see constantly in a lot of different scientific studies about how plants can thrive in the spaceflight environment. So these studies basically give us an insight into how plants are adapting to this unique environment. So when I started my postdoctoral studies, my doctoral studies, I started sleuthing through the data, and I noticed something else that was really interesting to me. What many of those genes that had changed their expression patterns in space, they basically had no known function. We have no idea what they're doing for the plants in space and why the plants are using them to physiologically adapt to the spaceflight environment. So that basically became my obsession. I needed to find out what the function of one of these unknown genes was and why the plants are using it in space. So another great quality of mentors is that is the trust that they have in you to overcome hurdles and figure out things for yourself. So both Rob and Annalisa were super supportive. They essentially gave me free rein to answer this burning question that I had. And so, it took a long while, 5 years, to try and figure out the function of this unknown gene. I chased down many rabbit holes and, went through meandering ideas and eventually figured it out. So because I got to discover the function of a completely unknown gene, I get to name it. And so I named it OMG1. And this is, for the many OMG moments, good and bad, in the process of discovering the function of this unknown gene. So it turns out OMG1 is involved in regulating the reactive oxygen species pathway in plants. Interestingly, this gene doesn't do much for plants on Earth, but it was highly used by plants growing in space. So this is another example of how the spaceflight environment can give us insight into interesting discoveries that would be hard to unfold on Earth. All right, so now I'm going to shift gears and talk about another approach to studying plants in space. So for my postdoctoral studies, I've teamed up with a couple of brilliant microbiologists in the field. And I'm interested in investigating how the plant microbiome can be used to help us prepare for future human missions in space. How can we use beneficial microbes to help improve plant growth, plant yield, and also while understanding what are the possible pathogens that could bring up potential issues for plant and the crew. Right, so scientists and engineers at the Kennedy Space Center have developed a hardware called VEGgie, which is the Vegetable Production System, and it's used for growing plants on the ISS. There are many different hardwares that scientists use to grow plants on the ISS, and this is just one of them that I happen to be working with. So in 2014, VEGgie was installed in the Columbus module on the ISS. So here is an up-close view of lettuce and Mizuna plants growing in the VEGgie hardware. And astronauts have eaten the lettuce that was grown in VEGgie. Here you can see a group of astronauts gathered with smiley faces and eating the lettuce that was grown on station. So as we embark on our preparations to go to the moon and Mars, plants are basically paramount. We have to bring them with us. Plants are essential for food production, life support systems, and well-being of the crew. I find gardening very therapeutic. I have a garden and I try and grow as many vegetables as I can. So I imagine that this would be something that, the crew would really enjoy as well. So since plants play such an important role in our spaceflight missions, it's really necessary for us to find ways to monitor and ensure that plants grow well in this environment and that we have a successful cultivation. So just like the human gut, plants too have their own microbiomes. Many of the surface symptoms of plant health, like tolerance to environmental stresses and disease resistance, can be attributed to microbial activity within the plant microbiome. So take, for instance, diverse microbial activity in the rhizosphere, which is the area near the roots, can really increase the healthy root development. And this will allow for plants to have adequate nutrient and water uptake. And, the more diverse the microbial activity, the more plants seem to do better. All right, so how do we analyze the plant microbiome and its function on the ISS? So actually studying the microbiome requires sequencing the DNA, sequencing the RNA. And most of the sequencing machines are huge. They are really large, big footprint, heavy mass, and many moving parts. So there is a relatively new sequencing platform called the MinION from the Oxford Nanopore Technology Company. And it's basically the size of a candy bar. And so NASA has decided that this would be the platform for sequencing in space. So small, it doesn't take up much size, much footprint, much mass, and all you need is a laptop to run this. So the MinION is also relatively new at performing host microbial metatranscriptome investigation. So metatranscriptome analysis basically looks at the functional activity of the microbes associated with the host. And in this case, we're going to look at how the functions of the microbes are associated with the plants. So here you see a picture of astronaut Kate Rubins with a laptop and the MinION sequencer on the ISS, and they've used it to sequence DNA in space. So now what I've done basically for my postdoctoral studies is to develop a method of processing the samples so that we can look at what the microbial community looks like in the plants and what they are actually doing for the plants. I've also come up with a bioinformatics pipeline to do this analysis. All right, so with this pipeline, you can actually find out what the microbes are, what is in the community. And you can also find out approximately how abundant they are. So, here's just one example. You can see the bigger the circle, the more of that particular microbe is present on the plant. The other thing you can do also is to look at what the functional activity of these microbes are. Here you can see the microbes are engaged in environmental adaptation, cell growth. You can see that they are also engaged in signal transduction and many different types of metabolic pathways. So for instance, we see that a lot of them are involved in signal transduction. Signal transduction systems enable the bacteria to kind of sense and respond and adapt to a wide range of environmental stresses and growth conditions. So we can then also go in closer and look at individual genes that are involved in specific pathways associated with signal transduction. Those genes can tell us what we need to pay attention to. The behavior of those genes over time can also guide us to make important conclusions and test hypotheses. So if you look over here, you can see a cluster of genes that are associated with the MAP signaling pathway. Interestingly, the MAP signaling pathway is also involved in fungal pathogenesis. So with these kind of tools, we can kind of keep track of what is going on in the plant microbiome, and monitoring the plant microbial functions can help us detect early signs of distress in the plants. So so what's next? The one main thing that I'm working on is to try and automate this entire process so that it can be easily performed on station by the astronauts. I'm also working with another professor at the University of Florida, Dr. Andy Sugar, on developing a plant disease diagnostic system. So imagine if you're traveling to Mars, and during that 9-month journey there, astronauts are growing plants to supplement their diet. What happens if a plant that is growing on station or on the spaceship gets infected by a pathogen? How do we determine what is causing the infection? How do we mitigate such problems if we don't know what was causing the problem, right? So I think we need to figure out ways to try and answer these questions. These are really important questions that Andy and I are currently working on and trying to develop methods and tools to address. So I guess, my takeaway message for girls who want to get into the field of space biology is basically to follow your passion. Even though the path may not be linear and you never know, you basically, you just never know what opportunities will lead you. But if you're true to yourself, you know what you want, you work hard, seek out great mentors. They play such a pivotal role in developing your career, and they can guide you and help you achieve your goals. So yeah, with that, that's the end of my presentation. Thank you.
Thank you so much, Natasha, for sharing with us your childhood ambition. I love the story, how
Thanks.
you persisted with passion and grit, and attracted the right mentors, and especially, as a mentor sometimes, or mentees, that you named each mentor that really helped you where you are today. That is something very important, and part of success, to tribute those people that help you to pave this non-linear way. So thank you for that.
Yeah.
And congratulations for the OMG gene and a million.
Thank you.
Fantastic name. Thank you for showing us
Yeah.
how to grow plants in space, and good luck with all of the future researches that you're doing. So
Thank you so much.
thank
Yes,
you very much. We're gonna look back to questions later on in the third part of the show. Now, let me please welcome Dr. John Kiss. He is the Dean of the College of Arts. Do you want to stop your screen sharing, Natasha? Just stop the screen sharing, yeah. We can still see your screen.
I just put my screen up there.
Yeah, and it works, yeah.
Okay, sorry.
So John is the Dean of the College of Arts and Science at the University of North Carolina-Greensboro, and his academic appointment is a professor of biology. He has mentored independent research projects by undergraduates and has served as major professor for 14 MS students, 8 doctoral students, and 7 postdoctoral scholars. His research focused on the gravitational and space biology of plants, and he has published 118 peer-reviewed papers. Dr. Kiss has also been invited to present seminars based on his research at universities throughout the US and in other 12 countries. He served as PI on grants from NASA, USDA, NSF, on 8 spaceflight experiments on the space shuttle, the former Russian space station Mir, and the International Space Station. Welcome, Dr. John. We need plants to go to Mars. With your help. The floor is yours.
Well, thank you very much for that very kind introduction. First, I want to congratulate Natasha. Fortunately, I've known her through our professional society, the American Society for Gravitational and Space Research, and I learned a few new things about her. And I do think she's a very inspiring young female scientist. So it's really a pleasure to be on the same panel with her. So she also had some really nice themes that I'm gonna follow up on. So I'm going to talk about why we need plants to go to Mars, and talk a little bit about some exciting results from our recent spaceflight experiments. So since she gave a little background on herself, I was born in Hungary, so I'm also an immigrant to the United States, but I came when I was 4 years old. I'm also a first generation college student. So I'm the first in my family to go to college in kind of another country. So I really relied on mentors. I had some great mentors, uh, different twist than Natasha, but kind of the same theme, people who have really inspired me. So I've always appreciated this. So I think this is an important theme. But fortunately, now I've been able to sort of pay it back as I've been a mentor to students. Just out of curiosity, I counted up all the students you mentioned, and I think about 2/3 of my students have been women. And I don't know, I found that really interesting. And of course, some of them now are professors at universities. They work for industry and for NASA. So I'm looking forward now to Natasha. I know because she had great mentors. She's going to be a great mentor herself. So I think that's a really nice theme. Um, I've always been interested in space. I'm going to give away my age here. I was born in 1960. So my life is pretty much the space age. NASA was founded in 1959. And I remember as a young child, I was 9 years old in 1969 when we had the Apollo 11 landing, and it was really cool for a 9-year-old. I got to stay up very late. It was late on the East Coast of the United States, but I've always been interested in the space program. And it wasn't until I went to, after graduate school, I was looking at postdocs, and I found this postdoc that dealt with ground-based NASA research. And then later on, as an independent investigator, I did both ground and flight work. And as you mentioned, I've been involved with 8 spaceflight projects. I know these will go in the notes, but I kind of gave a longer version of this at TEDx Greensboro, and all those are available online. And that one was called a little bit different. Someday we'll go on Mars, but here I am standing with the beautiful Arabidopsis plants, the space plants.
I saw that TED Talk, John.
Oh, good.
Very little did I know that we meet later virtually.
That's good. So let me say, why do we want to go to Mars? Well, Mars could probably help us learn more about Earth. So Mars, it's either Venus or Mars. Right now, it's Mars is our nearest neighbor, and sort of the formation of Mars and understanding how Mars formed and the geology could really help us learn more about our home. Of course, we're doing a lot on Mars now. In the summer, there was this launch of yet another rover called Perseverance, and it's a really cool project. And there's a picture of it. So some people, I guess this might be more in a negative tinge, but some people think that our population is increasing. We're running out of resources. Sooner or later, we need to go to other planets. So maybe this is the next step. I think a really great sort of philosophical reason maybe is discovering life on Mars. Right now, the only place we know for sure that there's life is on Earth. Now, personally, I'm pretty sure there was or is life on Mars, but we don't know that for sure. There's probably life on some other these planets like Europa and Enceladus. But I think if we actually confirm life on Mars, it'll really be a profound change for the way we think about humanity. By the way, if we look at life on Mars, it's probably not going to be like the guy on the left there, the popular version of aliens. It's probably going to be more like the image on the right, it's probably microbial if it existed in the past or maybe now. So I think there are a lot of really compelling reasons. And, really part of it is this simple diagram here. So if we're going to be going on long-term missions such as Mars, really plants, and Natasha alluded to this, are really part of the solution. So this is a natural synergistic relationship, right? That's pretty well known. We have the plants. You could grow plants. You could use them directly as a food source. But also, a really nice thing is plants will give off oxygen. which the astronauts, in this case, two of my favorite astronauts, the Kelly brothers, they will use that. And humans, the astronauts, will give off carbon dioxide as part of cellular respiration. And of course, the plants need this. So it's a lovely relationship. There are all sorts of other reasons to include plants. But actually, I noticed Natasha had a picture of this. She had space zucchini in there. And there's a really cool story about space zucchini. I don't know if she was at the same conference when Don Pettit talked about this. So astronaut Don Pettit gave a talk about this. Astronauts love working with plants. They love working with plant experiments. And this was a story where Don Pettit was on the International Space Station for 6 months. And he decided to, he had some zucchini seeds. Now, I don't know if it was his lunch or if it was an experiment, but he grew this plant hydroponically. And it was, he called it space zucchini. It was kind of like his pet. It followed him around, and as a fun thing, NASA did a blog called Diary of the Space Zucchini. But the story here is these astronauts are in this kind of sterile environment, and even in low Earth orbit, they just love seeing plants. Natasha showed the picture of them eating the lettuce. That's great. They get to eat the experiment. They see something green. And I used to not think very much of this, but I really think there's a lot here about the psychological benefits of plants as well. So when people talk about going to Mars, and I'm talking about like the popular media, in general, these are the kinds of things that people focus on. Usually the first thing is like Elon Musk, for instance. He'll talk about a big, big rocket. And it's true, we need a big, big rocket. So we need advanced rocket propulsion systems. And there's a couple of candidates for that. However, even if we had that big rocket, there's all sorts of other issues, effects of radiations on humans and plants. So we're covered in Earth, we're pretty much protected from radiation. Even on the ISS, there's protection through the Earth's radiation belts that are pretty far out. But if you're talking about going to Mars, both in deep space, getting there, and once on Mars, there's very high radiation, and that's a big problem. Of course, there's effects on human physiology, bone density and muscle mass is lost. And then we need life support systems, which plants will be part of. But the thing I focused on is this: biology and reduced gravity. So we actually know a lot about plants in microgravity and low Earth orbit, because plants have been part of all of these all of these missions from the earliest Soviet and American NASA missions. We actually know, we need to know more, but we know a lot, but we don't know much about reduced gravity. What will happen to plants in reduced gravity? And I'm going to talk about this in terms of our research, but one of the things we discovered, this was in microgravity, and hopefully these plants are moving. This is a time-lapse series, not terribly elegant, but very interesting. So if you look at this experiment, which was done on the International Space Station, sent up on a SpaceX rocket, what you will see is these are little plants, little seedlings. You actually could see the leaves, hypocotyls, and roots. Natasha showed a higher resolution picture of this. But what's happening is there's red light is being applied from the side, from the left side of the screen. And we discovered this new red light response. It's really strong in the roots. You could see it in the roots. The roots are growing toward red light. You could also see it in the stem-like hypocotyls. It's a little less obvious, but they are growing toward the red. They're also nutating, which is they're rotating around an axis, and that's another interesting story. So we discovered this red light response. If you do this experiment on Earth, the plants will be insensitive to the red light. They'll just grow straight up. They'll nutate. So it was a novel red light response that we discovered in roots and shoots. So let me get back to the reduced gravity. So what we know about this is gravity is attraction between bodies. So you could describe this in many ways, but the simplest way is on Earth, we're at 1G, so we're very Earth-centric. So we know about microgravity, like the astronaut on the left. There's essentially this condition of weightlessness in deep space flight or in low Earth orbit. But let's look at other bodies, and now NASA is very interested in this. We have Mars, which is a smaller planet, and so the gravity level on Mars is 3/8 G. The gravity level on the moon is 1/6 G. How do plants respond in this environment? And this is one of the things we focused on in one of our experiments on the International Space Station. And we actually know a little bit about this. If you look at those pictures from the late 60s, early 70s of astronauts on the moon, they weigh 1/6 what they do on Earth. So they're hopping around the moon, realize they have like a 100-kilogram backpack on. It's really hard to hop around, even if you're physically fit, with a 100-kilogram backpack. So we know about this. So this was one of the questions we asked. So one of the nice things about the space station is we have some centrifuges on board, and we used a facility that was designed by the European Space Agency. As an aside, 7 of my 8 experiments were collaborations between NASA and the European Space Agency. So in this last project, we worked with, my collaborator was from Spain, but we worked with groups in France, Netherlands, Germany, and Norway. In any case, there are these centrifuges on the space station, and they could create gravity or artificial gravity. So we made very good use of that. What we did was we did this experiment in microgravity, which I just described. So you could see that the stems and the roots are growing toward the red light coming from the left. If you do this experiment at the moon level of gravity at 1/6 G, it's the same result as in microgravity. So what happens is that you will get curvature toward red light. If you do the experiment at the Mars level of gravity, you increase the centrifugal speed, centrifugal force generated. There is no response to red light. They grow more or less straight. I did it the wrong way. So basically, the result is the same as on Earth. Now let me extrapolate this a little bit. And here's the simplest graphic. For plants, essentially the Mars level of gravity is just as good as Earth, at least for the parameters we studied. The moon level of gravity really isn't enough. So what this means is if you are going to be doing an experiment, if you're going to have greenhouses on the moon and Mars, there's all sorts of problems in growing plants. I'm not trying to sort of sidestep this, but one of the problems is on Mars, we're expecting, based on our results, that plants will behave like they do on Earth. On the moon, it's a different story. They're going to behave like in microgravity. So this is something we need to take into account. And another thing here is I like to leave you with this thought that we could use the International Space Station as a test bed for Mars. We still have it. There isn't a big centrifuge on board, but there are a number of small centrifuges. And we should really carefully look at the effects of whatever we can, whatever parameters of the moon and Mars levels of gravity. And since this is given the group that I'm speaking to, I was thinking of inspiring women I've met in terms of the space program. And one of the most inspiring women, and I think kind of an unsung hero, is Eileen Collins. Now, she was a space shuttle astronaut. She was not the first woman in space, but she was the first pilot of the space shuttle, the first female pilot of the space shuttle. And there's a lot of reasons it took a long time for this to happen, but she was the first pilot. She was pilot on 2 missions. and commander on another two. She was a phenomenal pilot because one of her missions was STS-115, which was the first mission after the Columbia disaster. And they really wanted an astronaut they could trust 1000%, and she was the one. So I urge you to read about her. I put a little web link there because I just don't think she gets enough credit. So thank you for listening to me. Thank you for inviting me.
Thank you very much, John, for your amazing presentation. Thank you for holding space for us women. And congratulations, you have a really high rate of mentoring female women, 2/3. That's amazing. Thank you. I know we now have 14 minutes to do some Q&A. I have a few questions here. And one question Simon asked, how do plants sense and react to gravity, and what are the molecular mechanisms involved?
You want to take it, Natasha, or do you want me to?
Yeah, well, I could give it a shot and then you can give your input as well.
Sure.
So molecularly, it looks like plants, I mean, they generally do well. They can grow in space. They change their genes expression. A lot of them looks like they change their cell wall modeling genes. They have a lot of responses to reactive oxygen species. And now reactive oxygen species can, it's a very ubiquitous molecule. It can help them sense the environment. It helps them cope with stresses. So, you know, these are some of the things that we are starting to realize how they are adapting to the spaceflight environment. There's still a lot more we're trying to figure out, you know, in terms of, I know Rob and Annalisa are working on trying to figure out the methylation, how that changes the gene expression and all sorts of stuff. John, you want to add on to it?
Yeah, I think that's a good summary. But the only thing I will add is, like, in the early stages of gravity sensing is reasonably well known, and they work by this mechanism called statoliths. They're these dense plastids with starch. They're found in the tip of the root and the tip of the shoot. And the cool thing about it is the general theme is just like the sensing in plants is, broadly speaking, similar to animals. So animals sense gravity. The way astronauts sense it is through our inner ears. We have these dense crystals called otoliths. And when we move our heads around, they move the otoliths and they tell us which way is up and down. And this is why astronauts actually get space sick, or the majority of them do. They need to adapt because their otoliths are all over the place. So they don't know which way is up and down because there is no up and down.
Thank you, John. The next question, Denise asked, which new systems should be optimized for performing plant life or space farming?
There's various ways you could answer that question. So right now on the space station, there are a number of facilities like the Advanced Plant Growth Habitat. I think that's what Natasha's group has used. So there's some facilities on the space station. I mean, one of the things I find really interesting, I actually think I'm a big fan of centrifuges and creating artificial gravity for the reasons I said. And NASA has some of these facilities, but I've always found it interesting that the European Space Agency has been more interested in providing these centrifuge-based facilities. So I'd like to see that. And then if you're talking about like greenhouses, it depends if you want to grow a greenhouse on the moon or Mars. It's going to be very, very different. I mean, I think on Mars, for instance, the the greenhouses, ironically, are probably going to be underground, and the light will be piped in because the radiation is so intense on the surface of Mars. I think humans living on the surface, it's going to be a really tough fight. So anyway,
Yeah.
I don't know if Natasha wants to add to that.
So I've been kind of collaborating with a lot of scientists out at Kennedy Space Center, and I know that they are testing different hardwares. They're building upon and developing newer hardwares to help grow plants in this environment. So a lot of them are using hydroponics, passive water transport mechanisms. They are looking at aeroponics, nutrient, what's it? Yeah, basically a lot of different techniques. I think they're still developing those tech and those hardware to try and see what grows plants in space, the best, essentially. So a lot of investigation is still going on right now. Yeah.
Thank you, Natasha. Malak, do we have more questions from the chat?
Yes, we do. The question is, do you think a new pathogen on a space grew plant is more dangerous or harder to control and study than an Earth grew plant?
So it's a really good question. There have been research that talks about how, in this unique environment, microbes tend to, not always, but tend to have more virulence factors. And so they could be more pathogenic, not all the time, in some scenarios. So it is a really good question because we are trying to figure that out. We need to know what the microbes are on the plants on Earth, and then we compare them to what they look like on station. And we want to see, under what circumstances do normal commensal microbes become pathogenic. These are all questions that we are still, trying to investigate and trying to come up with good answers and good methods to mitigate if that becomes an issue. Yeah.
Thank you, Doctor. Dr. John, do you want to add something to the question?
No, I think Natasha has that. I mean, I think the topic is also under researched, then we need more research on it.
Yeah.
Next question.
Thank you. Next question is, it has to do with the 3D footprinting. And what are your thoughts on the 3D footprinting?
Yeah, I'm not very familiar with the research that is going on 3D printing food for the astronauts. But, it never hurts to find new ways and new technology to help feed the astronauts or, the colony when we get to Mars or on the moon. It's great to always have different a diversity of technology and a diversity of backups, in case something happens. It's always good to know what other alternatives you can use, I think, in my opinion.
Thank you, Dr. Natasha. Dr. John, do you like to add something?
Yeah, I think 3D printing is very useful in general for the space program, but I don't really see it being very useful directly for like the plants as food. I mean, that's sort of a problem, right? If you go to Mars and like you need a tool or something, or something breaks down, I think it's going to be part of the solution. So I think it's useful in general for the space program, but not directly useful for the plant biology part.
Thank you, Dr. Natasha. Thank you, Dr. John. I have one question, and the question related to your research, how this research can help humanity on Earth to realize food security.
Yeah, let me take a stab at that. I think, both the research Natasha presented and that we're doing in my lab have Earth benefits for sure. In fact, NASA is always very cognizant of that. When we do these annual reports, they explicitly ask us, what are the Earth benefits? And there's a lot of, if you take a step back, there's all these benefits from the space program. On the human biology side, bone density mass is greatly reduced at a very rapid rate in microgravity, and we have an aging population, so it's relevant to osteoporosis. The plant research, so one could view space as a stressful environment, as a stress environment, even though the plants grow. So understanding like stress-related genes, I think, have applications to human agriculture. Our environment is getting more stressful because of global warming, climate change. We have increased saline intrusion into soils. So what we learn about these stress responses in microgravity and space are going to really also help us understand plants in terms of agriculture.
Yeah, I agree. For understanding the plant microbiome, there's so much benefits that we are discovering with the human gut microbiome, and understanding how the plant microbiome works, how it's helping plant growth, can also have a lot of benefits on Earth. We can better understand more natural methods of improving yield, soil remediation, all sorts of stuff that we can stand to benefit when we do such research on station.
Thank you very much. Thank you.
Thank you, everyone. So this was an amazing panel. Thank you for your presentations about space biology and sharing your trajectory. I think both as immigrants, how you paved the way and made this career that gives a lot of faith for our current mentees from developing countries who are, of course, in this crisis, seeking jobs with a lot of rejections. I hope this gives you hope with Natasha's passion to persevere. And thank you, John, for holding space for us women. And thank you, everyone, for joining this amazing panel. Lastly, I would love to have our traditional group picture. So if you choose, put yourself on camera. And thank you, everyone, tuning in from all different places of the world. We will welcome you next Saturday for the diversity show with Professor Moriba Jah and Isar, Mrs. Cecilia Baudo. She's also a Space for Women UNOSA mentor, sharing about space diversity through space oneness. I think it's very much needed right now. Thank you very much, everyone.
Thanks for the opportunity.
Thank you. Stay safe, everyone.
Yep.